I wish I had this kind of walk-on music when I teach class. I guess my takeaway is that if you have two Schmidt Fellows in your lab, they force you to speak. Because I see a lot of my colleagues just hanging out, enjoying their wine, and having a good time. So I was thinking about what ties all of us in this room together. And I think it's that we're all obsessed with some kind of scientific question or technological problem that we're trying to solve. And my obsession started when I was 20 years old, which is now, sadly, a long time ago, when I picked up a book because it had a shiny cover, okay? And the book was written by a futurist named Ray Kurzweil called The Age of Spiritual Machines. And he predicted in this book that someday we would have brain-computer interfaces that would allow us to link directly to computers with our thoughts, to control our environment, enhance our cognition, and better understand how the brain works. And I thought, dude, that's pretty cool. We should make that. And I was very lucky because at the time I was an undergrad at Brown, where some of the earliest work on brain-computer interfaces, or BCIs, was happening. And we were sticking electrodes into the brains of monkeys and later on human patients and recording activity from the motor cortex and using that to drive cursors on the computer screen. And that was really, really exciting. But I also realized that that's pretty invasive and that it wouldn't scale because there are only so many electrodes you can stick into the brain before you displace the entire volume of the brain. And so I decided to make it my mission to develop ways to interface with neurons in the brain and with cells elsewhere in the body that would be less invasive and allow us to talk not just to a handful of cells, but to millions or billions of cells that are inside of our organs. That sounds fun, but it turned out to be a really hard problem and quite multidisciplinary, which is probably why I'm up here, because it involves physics. What forms of energy can get in and out of our bodies? Unfortunately, things like light won't do the trick because we're opaque. So we need to turn to things like sound waves and magnetic fields. And then how do we couple that physics to what cells are doing? What kind of materials do we need to introduce into cells to make that happen? And one of the beliefs I've had for a long time, and now I think a few people are starting to believe me on this, is that in the long term, it's going to be easier than trying to introduce some kind of inorganic materials or sprinkle them throughout our organs, that it'll be easier to introduce genes into cells using all the latest tricks of gene delivery and genome editing approaches, that where those genes will carry instructions into cells for how to make the necessary materials themselves, that then would allow these cells to interact with the outside world. Okay, this sounds pretty sci-fi, so I just want to give you one concrete example of how we're doing this right now in the lab. And this uses ultrasound, which is a very widely used form of imaging. Can't see you guys very well, but how many of you have had an ultrasound? Okay, I think those of you who don't have your hands up are wrong. Ultrasound was probably your first picture, you just don't remember. And that illustrates also that ultrasound is very safe. It can go in and out of tissues without damaging them, and it works by scattering sound waves off of materials that have a little bit of a different density or stiffness inside the body and forming an image. And so we said, how do we get cells to interact with ultrasound? And this is where we got very, very lucky to come across these very unusual proteins that evolved more than 1 billion years ago as flotation devices in photosynthetic cells that live in water. Okay, these cells need to float near the top of the water to get enough sunlight for photosynthesis. And the way they evolved to do that is by forming these really unusual air-filled proteins inside the cell. Okay, imagine a little ping pong ball, an actual ping pong ball, that is shrunken by a factor of 100,000 and made out of proteins inside of a cell. And because it's filled with air and has a lower density, it helps these cells float. Now, that evolved more than 1 billion years ago. And then about 125 years ago, some German microbiologists discovered that these weird photosynthetic microbes contained these proteins. And then just over 10 years ago, we were the lucky ones to discover that because these proteins contain air, they're really good at bouncing off sound waves so that we can image them with ultrasound. And when we take the genes that allow the photosynthetic cells to make these little proteins and transfer them into cancer cells or immune cells or microbiome cells or neurons, we can start to see these cells with ultrasound inside the body. Okay, so there's this one very lucky multidisciplinary insight connecting a piece of biology from 1 billion years ago with the physics of ultrasound that helped us create a new field. And I'm very happy to say that now my obsession is shared, willingly or not, by the 30 lab members that I have in my lab at Caltech and a number of other labs around the world that are applying this technology towards problems in cancer. How do cells metastasize and where do they go? Towards immunology, how do immune cells get to the right place where there's infection or disease, and what activities do they undertake? To the microbiome, the bacteria that colonize our guts, where do they live? How do they interact? What are they doing? And yes, even neuroscience. And I'm very happy to say that thanks to Schmidt Science Fellowship support, Josh Chen, I don't know, where is Josh here somewhere, in my lab is one of the people that is applying these technologies to use ultrasound to look at brain activity, to look at the release of neurotransmitters in specific parts of the brain. And Ah-Young Kim, who's my other Schmidt Fellow who's here somewhere, is doing complementary to use ultrasound to manipulate the activity of cells. So are we there yet? Have I lived out the dream that I read about when I picked up that book with the shiny cover? Not yet, but we've made enough progress to keep the obsession alive. Thank you.